But anyways, I wanted to start off by saying that I'm going to be couching what I'm saying with empirical resources, and I'll try to talk about them as much as possible and cite them as much as possible so that people can do their own reading and that they know that I'm not sort of pulling something out of the ether, even though I am wearing this nice cloak. And I did make a meme about pondering the orb. That is not my intent. So, anyways, so viroids, right? The first article I have here, and I might actually take my phone like so, and I may sort of share some of this information using the camera in some cases, but honestly I'll try to keep it like this as much as possible. Because actually I think this is reversed in the video, so that doesn't help anyone, I suppose. But anyways, I have here the International Committee on Taxonomy of Viruses. So ICTV is a really great resource for viruses and viroids in general. But since we're talking about plant viroids, they're great for that too. And they have a lot of information. They keep pretty up to date, in my opinion, on the taxonomy and classification of these replicons. So the summary. So viroids, I'll just say this freebase without talking about it too much with the text, although we'll go over it. So what is a viroid versus a virus? A virus has it has genes and it has proteins, right? And particularly, not all viruses, but a lot of viruses have this sort of a protein shell, typically called a capsid, and that helps keep it sort of stabilized, and it allows it to sort of like keep its genetic contents inside. And then, like in the case of a bacteriophage, which is probably the most archetypical example that people give, the little alien spaceship-looking thing with the isohedral and then the long sort of like neck. Sometimes they're smaller, sometimes they're a lot longer, and then they have those sort of like projections where they meet with a bacterial cell. They inject the genetic material and replicate inside the cell. So that would be a bacteriophage. Other viruses have capsids, but they allow them to be pulled into like a cell, and then that allows them to then be have the genetic material be taken out and replicated, and that's what the virus wants. So up front, viruses use these proteins to exploit sort of channels that cell biology would use for other sorts of things like nutrient transport or the transport of other sort of compounds and other sorts of things like that. So a viroid lacks this capsid entirely. It's just a strand of RNA, and it doesn't even encode for various genes, which is really kind of fascinating when you think about it, because this makes it kind of vulnerable to a lot of different things. It means that it relies on things like mechanical transmission and vectoring through insects in order for it to move from host to host. I guess that's also true for a lot of viruses as well, but they are even less protected. So it's also kind of perplexing, because then there's the question of, well, why are viroids so effective? How are they, if a virus needs these capsids and other, indeed other pathogens, right? Bacteria, fungi, that sort of a thing, water molds, various taxa that are pathogenic to plants, they have a hard enough time as it is, and they've got, they've got cell membranes, and they've got, obviously genes and proteins and things that allow them to be virulent and effective, and even they have problems sometimes. But viroids are still able to be useful, and the main reason for this is because they induce genetic changes, and they basically, because of their RNA structure, they're able to, when they get into a cell, they interfere with the DNA production machinery of the cell, essentially. And there's two main places where that replication happens, and we'll go over that here. But there's two main taxa of viroids, and one of them generally infects the nucleus, and the other one the chloroplast. And it's important to note that viroids, for the most part, pretty much entirely infect plants. They don't infect animals. Some people would say that hepatitis D is a sort of a virusoid or a viroid-like sort of replicate with its own shell. But I am not a virologist, so I'm not going to make that claim here. But it is a claim that I have heard before, and you can research that yourself as well. But that's kind of a technical thing. I don't think it bears much for this particular conversation, but it's interesting to consider when we're talking about like the evolution of viruses and how they come to be as effective as they are and that kind of a thing. So viroids, essentially, you can think of them as strands of RNA that just by their very presence mess with the production of DNA, because they mess and they exploit with the replication machinery that would usually take RNA and then use it to produce DNA, for example. And because viroids also have some of these long strands of code can also silence, or they can become fragments, and those fragments will sort of bind with and like I said, sort of hurt or silence some of the immune responses that plants have, usually for viruses and viroids. So that's why that's what allows them to be successful. So that's kind of a general sort of summary. Here I have a summary from the ICTV. They say that viroids are small, a few hundred nucleotides long, circular, single-stranded, non-protein coding RNAs or ribonucleic acids that replicate autonomously in higher plants. Some are pathogenic, but others infect their hosts without causing any obvious symptoms. And that's true. A lot of the people who are listening here are probably aware of Hop Latent Viroid particularly, and we will talk about that too. But viroids don't always cause pathogenic symptoms, right? So sort of the definition of a pest or a pathogen is something that causes sort of dysfunction in the plant. If it didn't cause that dysfunction, is it really pathogenic, or is it just an endophyte? Or is it just an endophyte? Is it only contextually that way until the plant starts to stress out through heat or overhydration or underhydration or something like this? Then that can sometimes cause even viruses or other microbes that would normally be more benign to have a parasitic interaction with the plant or some sort of deleterious effect on the plant. A good example of this is the Huanglongbing or citrus greening disease that was recently found to be massive problem, causing tons of problems in citriculture, affected by an insect, this little bacteria, this phytoplasma. It is actually not a pathogen. Usually the pathogens, through finding a way to get resources from the plant, they are parasitic in that way. They are either destroying the cells and drinking the contents, or they're tricking the physiology to give them resources by exploiting pathways that they would use for mutualistic microbes. But it turned out that the bacteria isn't doing anything to try to get resources from the plant. It just simply causes a stress response in the cells, and if you mitigate those stress responses in the cells, they don't have a negative effect. In fact, the symptoms go away. So in some cases, you get organisms in plants that aren't actually going to cause a problem in the plant, actually, even though they're a foreign body. So that's a thing to consider when we're talking about the microbiome too. A lot of relationships are contextual, and some relationships are typically benign all the time, whereas other ones are a little bit more capricious or malleable. Although really I only say capricious because we don't know the factors all the time. Anyways, that was a really long digression. Unlike the genomes of the majority of viruses, viroid genomes are not protected by a protein capsid, like I was saying earlier. Now they have an example here, potato spindle tuber viroid, which is a genus of the Pospiviroidae. So it's in the Pospiviroid genus, right? So that's a typical member. They give us an example here. They articulate the genome here, single-stranded, circular RNA, about 250 to 430 nucleotides, adopting a rod-like, quasi-rod-like, or branched conformation of minimum free energy with conserved structural motifs. So that's all a bunch of technical jargon to a lot of people. But essentially, I want you to consider And I might be able to draw a diagram here or show you one on one of the research reports that I have. But basically, sometimes they kind of look like this. This is a very, very simplified sort of a diagram. But so sometimes they have this kind of like sort of long code structure, and then they have this like little hairpin here that's actually very structurally sound, essentially, because of the way that the binding of the RNA like sort of is assembled, essentially, so it doesn't lend itself to being sort of easily taken apart by certain processes anyways, sort of like enzymes and that kind of a thing. And then other times it's sort of a branch thing where you have sort of like multiples of these all kind of branching out. So that's just kind of like a visual aid, I suppose, that I'm trying to give you there. Very, very basic one, but one nonetheless. So they're in the cell. They get into the cell somehow through mechanical transmission, or maybe an aphid pierces the tissues and then sucks it all up and regurgitates it elsewhere, or something like this happens. And so the RNA strand, right, the viroid is put into the cells or into the phloem, and then it finds its way into the cells. And when that happens, it already has this sort of formation, but as it starts to bind with the RNA, then you have that. So like you have like RNA polymerase, and you have other machinery that sort of takes that RNA code and tries to do something with it, and that's the fatal flaw. That's the problem with the viroid there, or at least that's one example of infection. There are various kinds. So there are some that are long strands, some that are circular. They're symmetrical or asymmetrical as well. So that kind of depends. And I hope that I have explained that kind of in a way that is sort of visually understandable. So under the genome organization and replication of this document, so you can go to ICTV yourself and follow along, or you can just look at the reading yourself. They say that there's no evidence that viroid genomes encode proteins. So instead, viroid RNAs contain structural motifs that are associated with functional roles. These structural elements, conserved among members of several species and genera, allow viroids to use host pathways to replicate and move systemically within plants. Some viroid genomes, like the Avsunviroidae, which is one of the major families, contain cis-acting hammerhead ribozymes embedded in the strands of both polarities and catalyze RNA self-cleavage during replication, probably assisted by host proteins, they say here. Unlike viruses that primarily parasitize the host translation machinery, viroids mainly parasitize host transcription by subverting either nuclear RNA polymerase II, like in the case of the family Pospiviroidae, which is by the way Hop Latent Viroid is part of this group, or a nuclear encoded chloroplastic RNA polymerase, which is what the other family uses, to recognize RNA templates. So that was a lot to take in. Here is a diagram that they give, and I am going to actually take the time to kind of show you. Hopefully it's not backwards. You guys can tell me in the comments if that is the case, but and hopefully you can actually see this. But basically, as you can see here, you have the asymmetric pathway of the Pospiviroidae and the symmetric pathway of the Avsunviroidae, right? And so you have the RNA here. You got one end and another end. I'm not talking about this too articulately, I suppose. But see, you have RNA polymerase II, RNase 3 right here. You have these hairpins I was talking about. This gets coded. You get this fragment. The translation RNA ligase here, the circular form, right? And then this is the symmetric pathway. It goes through this process here, Again, not very articulate, I suppose. But basically, both of these processes terminate in sort of this circular RNA, right? And then that gets, continues to be replicated. And, here, models for viroid replication, right? There's different polarities. That talks about the red lines versus the blue lines. So positive and negative polarity, right? So essentially these, like I said earlier, they sort of they replicate by taking advantage of normal processes in the plant that would replicate the DNA, actually. The RNA, right? But in order to make DNA, right? That is how the process is supposed to work. And another thing to consider is that it's interesting to consider this from an evolutionary perspective, because there's this concept called the RNA hypothesis, or RNA world hypothesis, which is this idea that a long time ago, some of the first replicating molecules, sort of like RNA, right, were things like viroids, perhaps, or like replicating molecules similar to them, and they kind of perhaps predate cellular life, or they existed at or around the same time. It's a very complicated subject, right? But it also makes sense because they're kind of like, there's certain viruses also are like this too, but they're essentially like fossils. They're kind of like RNA, or like replicating sort of like fossils. So they still are relevant, but a lot of plants have developed ways to silence them, ways to sort of adapt and defend themselves against the infection. Despite the fact that viruses and viroids sort of exploit these processes, they are able to escape that. They are able to defend against that to some degree. Now, in some cases, the plants can't do this, and in other cases, one species of plant is going to be susceptible. Perhaps even certain individuals in a population are going to be susceptible, whereas others are not. And it comes down to this very, very fine molecular genetic sort of crosstalk that happens. And of course, if a plant's already very stressed from natural occurrences or pest pressure or things like this, then perhaps it will be less effective. But in a lot of cases, sometimes viroids don't necessarily kill the plant outright, but they stunt its growth because they're messing with the basic building blocks of cellular replication and hormone signaling and things like this. So what ends up happening is that for example, with Hop Latent Viroid, the reason why you see stunting, lower terpene and cannabinoid biosynthesis, even like modeling and weird developmental growth, is because at the cellular level, what viroids do is they mess with and they interfere with this sort of code production. And when plants do have antiviral strategies, like I was saying earlier, they do have sort of RNA silencing and microRNA that they produce extracellularly. Somebody in the comments was talking about exosomes too, which is, I feel, very relevant to sort of this conversation. So they produce these sort of microRNA that are meant to sort of bind with and interfere in the same way that sort of viroid RNA does that as well. And so by triggering this immune response, obviously that in and of itself has defense and resource and growth ramifications for the plant. So even if the plant is able to fight off an infection, which is usually not the case with like Hop Latent Viroid, for example, then they still are using a lot of resources in order to make this achievement happen. And at the cellular level, you still wind up with a lot of damage despite all of that, right? So going forward, the biology. Under the biology heading, they say, the main biological features of viroids are their ability to replicate autonomously and infect their host plants systemically. So they are a systemic pathogen. They are not I'm blanking, but like an epiphyte. They are an endophyte. They are able to move through the plant, through the cells, through the phloem. These features discriminate viroids from viroid-like satellite RNAs, which are small, circular, non-protein coding RNAs whose infectivity strictly depends on a co-infecting helper virus. Some people have heard of the term satellite virus as well, but this is essentially what they are talking about. There are cases where so it's not like they're part of the same genome. They're different viruses or viral particles, but they are able to synergize in unity when they are all present in a host, essentially. And it's very complicated too, but essentially that's what they're talking about here. This aspect needs to be carefully ascertained before classifying a small circular RNA as a viroid, right? Because you really want to know what you're talking about. And when you're talking about such a simple structure, even a structure that is oftentimes, if you're not thinking about it, if you're not looking for it, and of course before like 1967, there were cases where people were thinking that this was endogenous genetic material, but in fact it was totally foreign to the genome of the organism. These RNA viroids and other sorts of satellite viruses. So you have to be really careful when you're doing these sorts of assessments as to whether or not it's exogenous or endogenous to the organism, and that can be very tricky to tell if you're not actually testing for it. Where was I? Yeah. So autonomous replication and systemic movement must be experimentally proven by inoculating the candidate viroid RNA into virus-free hosts, followed by recovery of viroid RNA from the systemically invaded tissues of the inoculated plant. And that's what I just described there. That's one way to do that. The time needed for the systemic invasion of plants, generally weeks or months, depends on the viroid-host combination. Experimental transmission to herbaceous hosts in the greenhouse is achieved by rubbing carborundum dusted leaves with crude extracts or preparations of total nucleic acids extracted from infected tissue. Alternatively, plasmid DNAs containing dimeric head tail cDNA inserts or their in vitro RNA transcripts may be used. So this is all talking about how you tell whether you have it or not, but I wouldn't really call that the biology, so I'm going to skip that part. Host range. Viroids have been identified only in higher plants, both dicotyledons and monocotyledons. Individual viroids may have either wide or restricted natural host ranges, so some viroids, especially those replicating and accumulating in the chloroplast, the Avsunviroidae that I was talking about, infect only one or a few related plant species. So sometimes they can be very wide, and other times they can be very narrow in their host range. So symptoms. Viroids may cause plant diseases characterized by symptoms on leaves like chlorosis, mosaicism, curling, necrosis. On flowers, so color streaks, flowering delay, or cessation of flowering. Fruits, including discoloration, deformation, and suture cracking, which I'm sure a lot of people know what that is without maybe knowing the term. If you've ever seen like the fruits kind of like become cracked, and there's like this sort of suture-like damage that occurs from the wound, then you know what we're talking about there. Tubers, so size reduction and deformation, like with the potato spindle, and on the bark, so like cankers, distortion of the bark, pitting, cracking, scaling, and gumming, and or developmental disorders like stunting and impairment of rooting. So they talk about the demonstration of pathogenicity here, using for the fulfillment of Koch's postulates, right? I'm also going to probably pass this diagram or this description up here. It's kind of more the same. I kind of already went over that. Movement. This is a question a lot of people ask about. Early studies showed that the long-distance movement of potato spindle tuber viroid, for example, people will reference this a lot because it's one of the first ones, or perhaps the first viroid that was ever described. So we have a lot more research about it. compared to other viroids. So it tends to be kind of like a model organism, like a Arabidopsis is for plants. In addition, viroids may be transmitted mechanically by growers working in crops or by grafting infected material, which are the two biggest ways that Hop Latent Viroid is passed, for example. Transmission by cutting with sap-contaminated tools may occur in the field, especially in woody hosts. In plants propagated via seeds, viroids may be spread through seed and or pollen, with the exception of tomato planta macho viroid, which has been reported to be efficiently transmitted by aphids, right? So I mentioned insects earlier. It's kind of ambiguous how much this happens, that viroids are colonized or are facilitated by insects. It seems like, unlike viruses, which, for example, aphids are, in some ecological texts I've read, they're estimated to transmit 75% of viral transmissions themselves, or even up to 50%. So a pretty massive amount of just for aphids, not including other hemipterous insects that have like a piercing, sucking mouth part. So, they transmit a lot of viruses, but viroids seem to be a little bit less mobile on that, on that transmission route, and it's sort of unclear why that might be. I think a lot of it has to do with the fact that viruses, because of their capsid, allows them to exist and even in some cases bind with the physiology of their vectors. And for a long time, it was actually not well known if viruses actually infect their animal hosts too, in addition to their plant hosts. And it seems like that is true for some viruses actually, which is very, very interesting. But a lot of the times that is not what happens. They just kind of sort of pass through the physiology. With the exception, I already said that part. Viroids are generally not transmitted by vectors in nature, according to Skorick in 2017. Relationships among viroids. Nuclear replicating viroids, the Pospiviroidae, have been reported to be phylogenetically related to chloroplast replicating viroids, the family Avsunviroidae, and to viroid-like satellite RNAs. The apparent monophyletic origin of these infectious circular RNAs has been questioned. Several features of viroids support their possible origin in the RNA world, which was the hypothesis I was talking about earlier, including their small size, high G+C content, which would increase the fidelity of primordial RNA polymerases, circularity, which would eliminate the need for a specific initiation site during replication, the absence of protein coding capacity, and the presence in some of them of ribozymes, typical features of an RNA-based world. So what do they mean by that? So basically, what's a ribozyme? So for a long time we thought that ribonucleic acid was just some sort of coding, sort of genetic code, right? And that's kind of all it did. But it turns out that in the case of ribozymes, they can actually work like sort of like enzymes, like I was saying. And that's the main thing that makes viroids so pernicious and difficult to deal with, because essentially, due to their structure, they are actually able to bind, silence, and fragment RNA. So that's obviously going to that's literally like throwing a monkey wrench into the replication process of a cell and all the various organelles and DNA and that kind of a thing that's being produced. So RNA can do this and has a sort of enzymatic quality that was not known always. So that was more recent after RNA was understood. And that is what allows viroids to be as difficult as an adversary as they are. Actually, I'm going to take this time to look at some comments here, in case anyone has some questions here. So, Swello Vivo, yes. So that genetic material, if it's exogenous, it would be a virus or viroid, and endogenous, an exosome. That would be one example, yeah. That's how I understand it to be off the top of my head. Let's see here. Yeah, if you have any questions, drop them in the comments here. Plant demons, yeah, I would agree with that perspective. Oh yeah, thanks for letting me know that it is backwards. I could try the back camera potentially. That, you know, maybe I'll try that right now. Tell me if it. We are all learning together here. Can I do that? Yes, I can actually. Oh, look at that! Look at that. So you can actually show it. So let me go back to that diagram in case people were interested or curious about that here. So sorry about that. That ignorance is deafening, I'm sure, for people who are familiar. But yeah, so here's the asymmetric and the symmetric pathways there represented in the text literature. So thank you for that. That's why you check the comments. Oh, Simply Admin says, why do you refer to them as mechanical? No, I was referring to, I believe, in reference to that, I was referring to how they're transmitted. So it's mechanically transmitted. So like by touching, by physical interaction, that kind of a thing. So Cha asks, there are a lot of plants that have photochemicals that exhibit antiviral capabilities for humans, elderberry for example. Are there any instances of antiviral phytochemicals in plants helping plants? I think so. Yeah, absolutely. But I don't know any of off the top of my head. And if you're talking about like directly antiviral, nothing comes to my head currently. But certainly in an indirect way, a lot of compounds sort of help as a lot of actual molecules in plants from an immune perspective. They end up being they can have direct effects on like a potential pathogen like a bacterium or fungi or something like this, and they're a signal, so they can kind of do both things at the same time. Not only can they be toxic to a select group of pathogens or other microbes for that matter, but they can also actually signal and kind of create this cascade in immune response. And so it's sometimes hard to parse out that interaction. This question, would using an easy cloner be a way to transmit Hop Latent Viroid throughout your genetic stock? Potentially, if you're cutting into the phloem and and there's if there's this sharing of plant juices and phloem, then yeah, absolutely that can happen. Yeah, okay. So I'm going to leave this comment, curious about the liquid-to-liquid transmission, as all I have seen in papers about roots containing a higher volume of viroid over other parts, but haven't found much research or confirmation. That's very interesting. So that's actually a really important point. We don't always know, like in some cases viroids might have a higher titer in some branches or some parts of the plant over others, as either a dynamic of how they move through the phloem, or because certain tissues are more susceptible than others, or it could have to do with the state of infection and how that's affected the physiology, which could perhaps bias the physiology post-infection to concentrate in one place over another, whereas in at the very beginning that bias might not exist, but kind of like a sort of a niche construction sort of thing where, like for example, an organism builds a nest and builds many nests and tunnels, and over time that actually physically changes the environment in which their offspring are produced and which other organisms exist. So that can certainly occur, or in the DWC method as you're putting there. I appreciate the comment, wood glue. So, transmission. Viroids infecting horticultural... Oh wait, I'm sorry. Did I actually do the movement part as well? Oh, okay, I think I went on a tangent again. You'll have to apologize for that, or I'll have to apologize for that. You'll have to forgive me for it. Um, with the same, so I said that the early studies showed that long distance movement of Pospiviroidae takes place through the phloem, with the same route assumed to operate for all viroids. This route is also used by most plant viruses. Viroids also move within the cell to enter and exit the organelles in which they replicate and accumulate, and over short distances from cell to cell via plasmodesmata. Several structural motifs likely to interact with host proteins have been implicated in viroid trafficking, and there's a few references for that, which is one of the best things about these articles. Moving on to transmission, viroids infecting horticultural species are transmitted mainly by vegetative propagation of infected plants. In addition, viroids may be transmitted mechanically by, I already read this, I'm so sorry, by sap-contaminated tools that may occur in the field, especially in woody plants. Right, so my mistake there. Sorry about the reprint. I already mentioned about the relationships of these, relationship with other taxa. Okay, so viroids share their small genome size, circularity, and compact secondary structure with viroid-like satellite RNAs that, akin to some viroids, also contain what are called hammerhead ribozymes, like I was talking about earlier. However, infectivity of viroid-like satellite RNAs, sometimes named virusoids, is strictly dependent upon a co-infecting helper virus. Bioassays showing autonomous replication are needed to conclusively classify a small circular RNA as a viroid. And family demarcation criteria, because I am pedantic, I will briefly go over this as well. This is a question about, what are viroids, and how do you know it's one group or another? How do we classify them in a taxonomy and a phylogeny, right? Viroids are classified into families, two families, the Avsunviroidae and the Pospiviroidae, on the basis of their biological, biochemical, and structural features. Members of the family Avsunviroidae can form in the strands of both polarities, the hammerhead ribozymes that mediate replication in chloroplasts in which these viroids accumulate. So that entire family, all of those viroids, they colonize the chloroplasts of the plant. Members of the family Pospiviroidae lack hammerhead ribozymes but contain a central conserved region, CCR, in their rod-like or quasi-rod-like conformation and replicate in the nucleus, wherein these viroids also accumulate. And the Pospiviroidae are what the Hop Latent Viroid family is. They're a cocadviroid in the Pospiviroid family. And so that was that article. Kind of a slog, sorry about that if that was a little bit dry for everyone here. Let me get to some more sort of Hop Latent Viroid centric stuff, since I know a lot of people are curious about it. Here I have a paper. This is actually from I think a book, chapter 44. It says here, geographical distribution of viroids in Europe. They go over a bunch of different viroids, about their physiology, about the research about them. But in particular, so they have this part here. They talk about hop viroids. They say this is, I think, hop stunt viroid, I believe, has never been reported in Europe in hop, Humulus lupulus, right? Whereas hop latent viroid genus Cocadviroid was first identified in hop in Spain. And according to the paper, which I believe this is the reference to the actual identification, that was 1987, and Germany in 1988. Despite its name, it was also found in the United Kingdom in hop plants that showed low cone yield and reduced quality for the beer industry, and that was in 1990. Subsequently, hop latent viroid has been widely detected up to 90 to 100% of the tested hop germplasm in Belgium, the Czech Republic, France, Hungary, Poland, Portugal, Russia, Spain, and Serbia, and this was in 2003. Citrus bark cracking viroid has been found very recently to cause severe stunting of hops in Slovenia. It is also currently on the EPPO alert list, so that's the European Plant Protection Organization, I believe. And they have here in this table, 44.1, the geographical distribution of viroids in Europe. And if we go down here to Hop Latent Viroid, right down here, yeah, we have Belgium, Czech Republic, France, Germany, Hungary, Poland, Portugal, Russian Federation, Spain, Serbia, and the United Kingdom. So these are all places where Hop Latent Viroid is known from, and I'm sure that it is actually a larger group than that. So that was a small little sort of description I wanted to bring up here. You can check it out. I will put links in the description when I post this up on YouTube. Let me just check in the comments again really quickly if you have any questions or something like this. Russ Brandon asks, is alcohol not a sufficient method to clean viroids off of the surface area of your tools? So alcohol I've been told does not work actually on the Hop Latent Viroid, for example. And specifically, and I think that that's technically true. I think that it's more so the case that if I remember right, certain grades of alcohol are actually worse, and actually, in fact, higher grades of alcohol are worse than lower grades. I think that the general consensus, this actually might actually be more true for viruses than viroids, actually, and I guess it has to do with the water content actually allowing it to be drawn into the proteins. But the consensus that I'm aware of is that a highly oxidating agent or a sort of something like bleach that actually denatures the genetic material is your best bet for cleaning the surface area of tools. Someone here brings up a peroxide, for example, for powdery mildew anyways. Yeah, so good in the comments, people are bringing up, here we go, sorry. Yeah, so bleach or Vircon S, there we go, that is very good, thanks for that roundtable selections. Could virus travel through root exodus? True. Or assuming you mean viroid, potentially they could. It's interesting that you mentioned that because the rhizoplane of roots tends to have this sort of extracellular molecular matrix of, like many people have talked about, sort of carbohydrates and enzymes and just sort of what's called environmental DNA or eDNA, sort of just like being released through the roots, through the border cells in particular, and certainly that can be a harrowing journey for example, endophytes looking to associate more closely with the roots. They have to have adaptations that allow themselves to either suppress the immune system when they get to it internally, if that's the kind of endophyte that they are, or if that's the kind of microbe they are, if they're an endophyte versus an epiphyte. And certainly I could imagine that it would also be a harrowing journey for, of course, pathogens that are trying to do this as well, but they have their own adaptations, and viruses and viroids potentially as well. Can you explain the mechanism of how antivirals like ribavirin reduce the viral titer during pre-treatment? Not very well, to be honest. I think that's the honest answer to give. But essentially, I could probably look it up here and explain it to you really quickly if it's not too difficult to understand. I may be able to interpret the answer without knowing it currently myself. So it says here, a quick cursory search on Google says that is a guanosine analog used to stop viral RNA synthesis and viral messenger RNA capping. This is on Wikipedia. Thus, it is a nucleoside inhibitor. Ribavirin is a prodrug, which when metabolized resembles purine RNA nucleotides. In this form, it interferes with RNA metabolism required for viral replication. So that makes sense to me. Basically, yeah, they sum it up really well. So it's an analog used to stop viral RNA synthesis and the capping of messenger RNA. So that's going to be, like I was saying earlier, that is one way to throw a monkey wrench into the synthesis of DNA or RNA, right? Is that you are essentially taking advantage of how the structures normally bind, and you're binding them with things that they don't mean to bind with, and that just screws it all up for them. And that's why that works really well. And actually, this is the same way that animal and plant antiviral responses work as well. They, or at least with regards to silencing or RNAi, which is RNA interference. So yeah, so hopefully that answers your question. Cheddar Bob has his mealybug under control. That is great to hear. Frankenseed's Monster says, I was talking to a tissue culture lab and they recommended bleach instead of ISO. That makes sense. Can you explain how nucleoside inhibitors like... Oh, I see. I already went over that. Yeah, so hopefully that makes sense. I've not personally used ribavirin for pre-treatment for tissue culture myself, so I don't know much about the actual sort of methodology associated with that, but that's why those inhibitors work apparently. Okay, so I'm going to move to this report here. I have a couple of these. One of them is I have two main ones that if I don't get to everything here because Instagram doesn't let me or whatever, I want you to be able to reference them yourself. So one of them is this advanced review by Charith Raj Adkar Purushottama and Jean-Pierre Perriot, probably, who knows? But the review is called Current Overview on Viroid-Host Interactions, and it was published recently in 2019, so it's very up to date. And the other one is called The Role of Phytohormones in Plant Viroid Interactions. So these two are big to me. I was looking over them and there were some of the inspiration for this video, so I haven't actually even gotten to the inspiration of this video. Swello Vivo asks, do you have any experience on curing viroid-infected plants? Not conventionally, no. Usually this is very difficult or impossible to do at the field level. Usually you have to do tissue culture because they're not typically they're not conventionally curable. But this might not be the case in the future. There might be more techniques. Maybe I'm not aware of them myself, or maybe the greater population isn't aware of them. Certainly I am not. But the example that I gave about the phytoplasma was very interesting to me because also, so not only was it found to not be really a pathogen, but it was also found to be very susceptible to, kind of like what they're saying here, sort of phytohormones that mediated cellular stress response and caused the reduction and elimination of symptoms from an organism that for a long time we thought was this deleterious pathogen. And I think that a similar sort of thing might be possible with viroids or even other plant viruses, something to accentuate the antiviral immune response of the plant. The problem, though, is that if they don't already exist, and probably also the application has to be done early enough for it to matter. And I think that might be the crucial linchpin problem, is that for a lot of these viroids, they tend, like for example, Hop Latent Viroid, they have viral latency. They are latent viroids, which means that you don't see the symptoms, you don't really know that you even have it until it's too late, until it's really colonized a lot of the plant tissues already. And that's kind of the issue, is that they're silent and deadly in that way. Yeah. And I agree that epigenetics could help us find cures and prevention in that way. I really do believe that there's a way that we could exploit sort of those natural physiological responses, but we have to understand what they are a little bit more sophisticatedly, and I don't think that we're quite there yet. But I have great hope, because that could lead to a lot of sort of like low-tech, kind of low-cost ways of dealing with the viroid. And potentially we might not be able to do it as much. Like with hops, for example, the way that people dealt with hops is that they didn't deal with hops. They developed cultivars that were resistant, and they developed an infrastructure that allowed for very, very high fidelity plant production that had these sort of resistances, and people rely on those quite a bit because of that. So that's quite a bit narrowed down. I'm not an expert on hops cultivation myself, but that's how I understand it to be the case. And I don't think the cannabis space is likely to go down that narrowing route. Maybe in some cases we will, but I think that a lot of people will ultimately want to keep their plants sort of viroid free through other biosecurity means, and hopefully this would be one way to do that. So going to the first one, the advanced review, there was a section that I wanted to discuss. Oh, this is a really great example here. So kind of that sort of structure that I was talking about here, you can kind of see what I'm talking about here, like the PLMVD here. You kind of have the branching that I was referring to in the article from before, the ICTV article. So you have some of them that are sort of linear, right, like the top one, and others that are sort of branched out, like the bottom here. And then they can change over time, depending on how they are, what stage of that they're replicating in, and that sort of a thing. So I really liked this table in particular. Here, I'll just show it to you. Now that you've introduced me to doing this, I think I'll be doing a little bit more. So this table 1 says characteristic features used for the classification of viroids, right? So the structure. So we've got the two big families, right? The Pospiviroidae and the Avsunviroidae. So on the left we have a rod-like. Terminal left, pathogenicity, central variable, and terminal right, that refers to the structural and functional domains, right? So ribozyme activity, no. Replication in this host is in the nucleolus. Replication mode is asymmetric rolling circle. And the enzymes involved include the RNA templated RNA transcription, DNA dependent, RNA polymerase II, and depending on, and the host range is depending on viroid species, either broad or narrow. So it could be either one of those things. But these are the enzymes that are involved in the replication of the viroid, of those families, of those genera in that family. But in the other family, we have a Y-shaped or branch form. We have a structural functional domains that are not applicable apparently, which is exciting. Ribozyme activity, yes. So these are the ones that are able to break and disengage some of the RNA code when they come into contact with it. They replicate in the chloroplast. They have a symmetric rolling circle style as opposed to the asymmetric one, and the enzymes involved in their replication include the RNA templated RNA transcription, nuclear encoded chloroplastic RNA polymerase, and they have a narrow host range. So that's that. So again, those are two main families from which a lot of these different genera descend, and Hop Latent Viroid would be one species that has descended in that way. And what else? else was it? I think there was another section here that I highlighted. Oh, right. So I don't know if this diagram helps anyone visually, because it is still kind of technical, but I found it useful. So for people who have maybe a similar kind of background as me, or have some like biology background, like some of the people commenting have here, you can kind of see this example of a mature viroid at the top, right, and the replicative intermediate here. So positive and negative polarity right at the top right. So host a messenger RNA, viroid snRNA interaction. So the VD stands for viroid, and I believe the S is small. So the viroid small RNA interaction. So as you can see, they bind with these coding regions, and the mRNA gets cleaved. You get the production of double-stranded RNA, and then the generation of secondary si RNA, which is I think that's small. Is that small interfering? Yeah, small interfering RNA, and then that induces the viroid symptoms, like here in the potato spindle tuber viroid. So you can see the control has no problem, and the experimented one that has the viroid very much has the viroid. You can see that stunting damage. So that comes from sort of cellular dysfunction. And here, they say here very plainly in a nutshell, the RNA silencing initiated by the viroid is multiplexed by the host's RDR6 to act on the host's own messenger RNAs, resulting in symptom expression. So what does that mean? It means that, like I've been saying this entire time, and I don't mean to be somebody who is sort of repeating themselves over and over again, but it really is that simple. They exploit the machinery of the cell used to create DNA, essentially an RNA for that matter, and organisms that do this, like viruses and viroids, have existed for so long that plants, animals, various organisms have their own sort of antiviral defense systems that are meant to kind of avoid this or counteract it, because it's a very exploitable system. All of this sort of production happening in various parts of the cell. On that note, you've even got cases where you get mutations, and you have, what's the word I'm looking for? You have like correction, enzymes and things that go around and sort of like correct if you have mistakes. So like read-write errors and error correction happens as well. So there are even organisms that are that selectively do kind of like what the RNA does to destroy and disrupt genetic expression and things like this. And it's not like it's a conscious action, right? There's not like there's this mind that is directing the RNA. It just happens to have, these structures that work really well. And in this particular report, they talk about the conclusion and future perspective about viroids. And I highlighted a few sections here. Basically, they say that demonstration of the accumulation of viroid small RNA in viroid-infected plants. Actually, before I go forward, I just want to kind of put this all into a nutshell. I think I've kind of got a zeitgeist idea of how to put it. Basically, think and remember that inside of cells, inside of the cytoplasm of cells, right, there is all of these organelles, there are all of these compounds being produced, there are all of these genetic code lines that are moving around and being produced and are in various states of production, kind of like an industrial production line. I think that's a pretty good metaphor for it. And they're all kind of buzzing around, right? And there's redox reactions as well going on. There's different charges, that are on the cellular membrane, or in the case of plants, like the cell wall and the plasmodesmata. And so there's all this free kind of like genetic material flowing around kind of doing their own thing. And it's kind of like it's like lighting a match in a fireworks warehouse, right? Like there you can very quickly introduce a ton of chaos to this already very chaotic system, and that's how you get things like mutations, and that's how you get things like virulence for viroids and viruses when they get into plant cells. So just kind of keeping that in mind, I'm going to continue here. Demonstration of the accumulation of viroid small RNA in viroid-infected plants and the possible role of the host RNA silencing machinery led to speculation of the direct and or indirect involvement in viroid small RNA in viroid-induced disease symptom expression. The recent in planta experiments aimed at defining the viroid small RNA-induced RNA silencing of host messenger RNAs helped define the viroid small RNA-induced regulation of the host's normal gene expression in a sequence-specific manner, and could explain why a minor sequence variation between viroid strains could induce an array of symptoms in susceptible host cultivars. So again, you can have a cultivar of a plant that will have a totally different reaction to a viroid, and this is my hope that this is the case with the hop latent viroid in cannabis, and indeed also with hops for that matter. They were able to breed resistance, so there's a likelihood that we could also breed that same resistance or exploit the same machinery that allows us to do that as well, although that would probably be a bit of a ham-fisted response, because it's not nearly as sophisticated as having sort of the biological system that's able to do these things itself immediately on detection. And that detection is very important. They say that despite viroids' lack of any protein coding capacity, studies have shown that viroid infection could either affect the host's translational machinery or could regulate translation. So obviously, if you're able to get into the cell and alter this machinery and change how the cell expresses itself and how the genes express themselves, and this is going to very quickly, depending on what things are affected, like metabolism, defense compounds, immune signaling response, all of these things, even throwing a small little wrench into that machinery can have massive downstream effects as the cell tries to replicate and forgets how to do so, and literal aspects of the physiology are going to be either non-existent or at very low levels because of it, right? Thermodynamic folding of sequenced viroid RNA has helped in understanding the two-dimensional structure of the mature viroid molecule for many years. They're talking about kind of how the structure of the viroid is sort of folded and collapsed on itself like proteins are, for example, already. They say that this could elucidate what permits the mature viroid to escape the host's RNA silencing machinery, even though viroid infection triggers the host RNA silencing, right? Yes. So so it's kind of unclear to us why it is that if plants do have this RNA silencing technique and this ability, then why is it that viroids are able to escape it? Because they are just strands of RNA after all, but they are a little bit more complicated than that, seemingly. And I think, again, it's very important to consider the context of all of the different things that are coming across and happening during just normal cellular processes, right? It is a very chaotic space. So that was the review. I only took a couple of sections out of it, but I implore people who are interested to look at the research themselves. And here in the role of phytohormones and plant-viroid interactions, I didn't actually get through everything here, but I wanted to go over again some of these points that they make here. This paper was produced in, or I think this is a chapter from a book. Yes, it is a chapter from a book. This is from Applied Plant Biotechnology for Improving Resistance to Biotic Stress. This was copyright 2020. But if you're interested, I will send it to you, no problem. So you're probably wondering what is RNA silencing and all this sort of a thing, and this is why I kind of highlighted some parts of this this paper, because they do talk about this sort of phytohormonal technique that plants utilize. So RNA silencing plays a role in modulating many of these interactions with viruses and viroids they're talking about here. Viroid infection often results in widespread changes in host plant cell functions involving defense-related pathways, signal transduction, hormone balance, and energy resources. So even just the basic metabolism gets messed up quite a bit. The mechanism of viroid pathogenesis is mediated directly by the viroid genome itself, or by viroid genome-derived single or double-stranded RNAs, and that expression of symptoms is a result of systemic infection, may be an outcome of direct interactions of viroid-derived small RNAs with so far unknown host plant factors, either in the organelle, where the viroid replicates, or in the cytoplasm, where they accumulate during its movement, and then of course out into the phloem and into other cells. They say here that 3 general pathways account for this mechanism influencing host gene expression. So viroids, basically like a lot of other organisms, both sort of mutualistic and parasitic, whether they're fungi, bacteria, oomycetes, or in this case viruses and viroids, they are able to elucidate genetic responses, genetic changes, or they're able to actually modify genetic expression themselves. This is one of the reasons why microbiome dynamics are so dang complex, because it is this sort of like communication crosstalk between the microbes and the plant host and other organisms that are involved as well. And so they're happening sort of all at the same time, sort of complexly. And I know that we paint a lot of mutualisms with plants as kind of hunky-dory and very sort of happy, but it's actually a lot less free and kind of like happy as it is sort of like a in a lot of cases I think that it might be more accurate to call it almost like a jailer, or perhaps some sort of a military commando kind of taking the microbes and walking them around and being very strict and very closely guarded. In a lot of cases, like with rhizobia, the plant's immune system is very active because even though it's a foreign body that's giving them resources, like in the case of fixing nitrogen or inorganic phosphorus or other sorts of things, they're still a foreign body, and they have to make sure that the symbiont that they've interacted with is actually giving them what they need and is not exploiting them for more resources or anything like that. And in a lot of cases, this sort of immune response can cause the relationships to not form in the first place. And so in that way, there's sort of this selectivity. So anyways, again, a digression, I'm sorry. But hopefully that helps paint sort of a picture for you when it comes to these interactions. So symbioses are constantly doing this, modulating their host genetic expression and that sort of a thing. So if you aren't already aware of that, that does happen all the time to plants. And of course that's going to cause issues and changes and stressors on replication of cells and that kind of a thing. So not all symbioses are good, and certainly not all mutualisms are created the same. So the 3 pathways that account for this mechanism of influencing the host gene expression: A, mature viroid RNA host factor interactions via induction, activation, or blocking protein functions. B, viroid double-stranded RNAs processed by specific dicer-like enzymes. Now, dicer enzymes are used to cleave RNA or DNA, or genetic material. So dicers are really important because they can do a lot of this like silencing that I was talking about earlier, and creating like fragments of genetic material. So they process by specific dicer-like enzymes into small RNAs, like I was saying, fragments, right, which guide the RNA-induced silencing complex, or RISC, R-I-S-C, which is host messenger RNA cleavage or host DNA methylation. So people familiar with epigenetics know that methylated genes sort of alter how the genes are expressed. And that's one way that epigenetic expression happens. And transcriptional silencing. And C, hormone-mediated responses, though altered plant gene expression through altered gene expression and endogenous microRNAs. So like viroids, certain viroids anyways, and I think they're talking about the Avsunviroidae, that family that has the hammerhead ribozymes. They're able to just sort of slice and dice this RNA and create these sort of fragmented RNAs that can go around and bind with things and really muck it all up. Here's just a few excerpts from the paper. It looks like an interesting comment here. From a microbiome perspective, virus could have played a huge role on genetic adaptation for animals and plants. Are there beneficial viruses and viroids? Yes and yes. And I posted a while ago about Luca, the last universal cellular ancestor, and also about its virome, and how it's important to know that one of the reasons why we think viruses have been around for so long, and one of the reasons why it seems like it's the case, is because viral genetic information is found in cells, right? And is found in very ancient, very sort of primitive primary machinery. And certainly they and other pathogenic sort of organisms had massive selection pressure on the cells that they evolved, multiple billions of years ago. And here we are now, we still have this sort of interaction because of, and actually I recently read a paper that I really enjoyed it because it talked about viruses, it talked about microbiome dynamics, they talked about what are called mobile genetic elements, or MGEs. And if you're interested in that subject, you should definitely check it out, because that is one of the ways that horizontal gene transfer happens so readily, is through viruses and maybe not viroids as much, but through viruses and bacteria, and the sort of the genetic information that sometimes gets inserted into cells and stays into the germline, and that does happen. Yeah. Oh yeah, and I've also talked about cannabis cryptic viruses as well. So that's very interesting. That kind of stuff just happens. Yeah. Mitochondria theory on how that became part of our evolutionary relationship. So that's endosymbiosis theory, and I recently posted about that as well, which is pretty interesting. So those two posts about Luca and the endosymbiosis theory is really interesting. I would definitely look into it. It's very, very, very exciting stuff. So plant hormones usually do not regulate defense responses via a linear mode of action, but through a complex network of regulatory interactions involving various developmental processes and defense signaling pathways. Like, anyone who's ever used hormones know that the dose is very important. I guess whether we're talking about things with animals or with plants, right? There's this, and I'm not an expert, but there's this dose-dependent response with plants, right? Where if people apply too much gibberellic acid or auxins or cytokinins or things like this, then you could have the opposite effect of what you were trying to go for, which is kind of counterintuitive. You would think, if you put more of a certain compound or a hormone, then you get more of that effect, and if you put less, you get less of that effect. But actually, it's way more complicated than that, because of course, plants have other hormones they're already producing endogenously when they're developing, and so those interact with each other. That's why I'm so pedantic about immune eliciting techniques, whether we're using microbes to alter the hormone production or terpene production of plants, or whether we're using the actual compounds themselves, or we're using like something that induces systemic acquired resistance and that kind of a thing. Because when you do that, you are eliciting an immune response. And when you elicit the immune response, what we've found is that this can have negative effects on downstream effects with the plant defense and immune response, and it can interfere with other immune responses that might be happening at the same time, or might happen in the future. And depending on what organism is actually colonizing the plant, whether it's like a piercing, sucking mouthpart insect, or an herbivorous insect that has gnashing mandibular feeding style where it chews rather, those will elicit different responses. And also with different pathogens, biotrophic pathogens like powdery mildew versus and also, for example, maybe viroids even would come into this sort of qualification, versus necrotrophs or hemibiotrophs that are able to, they don't play and communicate with the physiology to convince it to give it resources sort of forcefully. No, the necrotrophs will just, like botrytis, they can switch to this form and they'll just destroy the cells. They'll exploit and elicit the hypersensitive response that kills the cell, that basically elicits programmed cell death. They will elicit that on purpose, and then they'll suck up the nutrient-rich contents. They literally want the plant cells to do what they're programmed to do in that way, but that obviously doesn't help the plant. So the plant has to know which system to use, and how it achieves that is through this sort of soup of hormonal signal cascades and things that can last hours or even days after they are elicited. And some of those processes have to be, or some of those compounds and elicitors have to be recycled. And if they're not given the ability to recycle, or if we're constantly eliciting other immune responses that do the diametrically opposed thing, then that causes a problem, and that can cause an issue with your immune response. So just be careful. There's a reason why it took, for the fourth time now, I think, in this conversation, to sort of digress and talk about this sort of niche subject, because it doesn't get talked very much, and I know it's not very popular to get super complex in the weeds, but it's important to know that, because if you're buying products and expecting a result, you should know how those results happen, essentially. And that's kind of what we're talking about here with viroids. So further, using tomato, potato, spindle, tuber viroid interaction as a model system, it has been shown that viroid infection alters the expression of host genes that encode products involved in defense, stress response, cell wall structure, chloroplast function, protein metabolism, and other diverse functions. So a lot of things. Increasing number of microarray and transcriptome studies provide evidence that different hormone signaling pathways are affected in response to viroid infection. Together, these results indicate alterations in almost all hormonal pathways. In this chapter, we will discuss on 4 of them, which would be the salicylic acid, jasmonic acid, BR, and auxins. BR, how am I forgetting what BR is? Let me just skip down here and see if they tell me. Oh yeah, brassinosteroids. Okay, yeah. So I'm not going to get into all of those. And that was actually all that I had highlighted here for this paper. But I'll go down to the conclusion. But as you can see, because hormones are such an important signal, so many things are hormones, so many things signal or indirectly or directly affect hormone signaling, that it's kind of, again, it's also another process it's very hard to parse out from other processes. But so much of plant processes are hormones, essentially. Sorry to be so circular in my description there, but that's just the case of it. So of course, if a viroid is affecting all of these different genes and their expression, then naturally these hormones are going to be affected as well. And if those hormones are affected, then the way for the body of the plant to sort of communicate with itself and regulate itself is going to be severely disrupted. And that is the kind of result that you're seeing. That is why like Hop Latent Viroid stunts hops and cannabis, for example. Yeah. Have you ever identified exosomes that act as viroids? No, not personally. That is not research that I conduct. So conclusion here, they say that although it is generally accepted that viroid diseases are induced by specific interference with regulation of host gene expression, the specific mechanisms triggering the onset of symptoms still remain hypothetical. Honestly, it's very difficult to research these viroids, and when researchers are doing so, they are very careful in how they talk about it. They're very careful in deriving conclusions from model systems and from experimentation until they're very sure. They have to use a lot of corroborating independent evidence to be sure that what's happening at a genetic and molecular level is actually happening, and not just the abstraction of it. A very good example of this is like, just even how we use models to describe things like chemical structure, like the chemical skeleton that everyone has seen. Like obviously atoms and molecules, they aren't really connected exactly like that, right? It's not mechanical like this. There's a lot of much more ephemeral interactions that are happening with electron shells and atoms and vibrations and all of that. So that's an abstraction that we use as a mental shorthand for us to understand the world as we do. But obviously that's not exactly what's happening, and that's kind of what they're describing here. Going forward, according to current data, it appears that the role of salicylic acid, jesmonic acid, racinosteroids, and auxin signaling pathways in plant responses to viroid infection depends on the host-viroid combination. Although the importance of phytohormone in the regulation of plant immunity is evident, obviously, current knowledge is not sufficient to establish a general model of their action during plant viroid infections. Given the complex network of phytohormone signaling pathways, further efforts should be made to elucidate the role of each phytohormone in the expansion of host responses and the development of symptoms in plants infected with viroids. Right? So that makes sense to me. Obviously, more experimentation is necessary. On the basis of recent advances in transcription profiling, it is expected that many more hormone-induced genes will soon be identified, facilitating a comparative analysis of promoter sequences and subsequent identification of putative hormone response elements. So we have ideas about what some of these genes are, how they affect the hormones themselves, but we don't really know in great detail, and people are, responsible researchers are just kind of saying this outright. We know that there are these things that are being affected, we know that that must be what's happening, but we don't know in great detail how these interactions happen, because they are very complex and complicated. Like I was describing with cell biology, like all of the various things that are happening in the cells, and parsing that all out is difficult to do. And the more you add that complexity, the more difficult it is to do that. By combining different approaches, it is necessary to further investigate whether a particular phytohormone activates or suppresses the defense-like responses, and how phytohormones together orchestrate these responses at the level of the entire plant, as well as in particular organs. More time-scale experiments may help distinguish the changes that are part of the defense signaling pathway from those associated with symptom development. Deciphering how phytohormones act and interact, and characterizing new players in signaling networks is crucial to understand how some host species survive viroid attack. New findings could help the development of hormone-based breeding programs aiming to improve plant resistance or tolerance to viroids. And that, I think, is a pretty good conclusion to kind of our conversation here. So the answer is, do we know much about viroids? Yeah, we know a lot about them. Do we know everything there is to know? Of course not. Do we know some of the finer details of how they affect what they affect and what they affect? Kind of. We know that they exploit genetic expression. We know that they exploit hormone signaling pathways. We know that they are obviously RNA themselves, and that different viroids can have sort of different colonization effects on their hosts. Sometimes it's incredibly deleterious, and in other times it is pretty benign and symptomless. And in those cases where they are symptomless, we're not really sure how or why that happens exactly, and how the viroid is still able to exist in the plant. I think it's important also at this point to continue to reiterate that unlike, for example, fungi or bacteria that are usually, so they're pathogens, right? So why are they pathogens? Because they cause physiological problems. Well, why do they cause those things? A lot of things happen. Sometimes they are mutualists that turn parasitic under certain stress conditions. Sometimes mutations happen in the genes that allow them to cause damage when they normally wouldn't have, or because they lose the ability to provide benefits to the plant and only take instead of give. In some cases that happens as well. And in other cases we have situations where the microbes, they just are able to exploit pathways that the plants don't want them, wouldn't want them to do, that are deleterious to the plant rather. And so in those cases, the pathogens might be essentially trying to acquire resources, or just happen to acquire resources in a way that damages the plant, and that's what causes it to be parasitic, right? There's a reason why it is the way that it is. There's something that's happening that causes it, and there's many different causes that can occur. But a lot of times it's in order to be a parasite that takes resources, and those resources are then in a sophisticated, refined way used to create like conidia or spores or more themselves, to colonize and then move to other hosts. But in the case of like viruses and viroids, they're not like getting resources, they're just replicating, right? And so for that reason, it's not like a viroid, when it goes into a plant, it doesn't like derive resources. Does that make sense? It's sort of like, it doesn't, it's not like a bacterium that might be trying to take in like a carbon-based resource or a nitrogenous resource and then use that metabolically to power its body and power its reproduction and power all of these things, right? That's the traditional thing that we think of for a lot of parasites, but viroids are parasites in a different way. They're not even trying to garner resources. They're just sort of replication agents that get caught up in a system that they can exploit, that happens to interfere with them as much as they interfere with it in a lot of ways. And that's just my I want to be clear here, that's just my kind of view on it. I don't know if that's shared necessarily with other people, in specific, like how I said it. But hopefully that kind of explains the difference between those kinds of parasitism, because as people who follow me know, I'm very, very specific about how I kind of approach these topics and how they are described, and how they occur the way that they do, because I understand what those sort of niche interactions are and what causes them as much as I can, at least. And here I'm going to see if people have any questions before I end the live stream. Yeah, Woodglue says its resource is a place for replication. Yeah, exactly. Essentially, that is the case. If you want to even go that far in describing a resource as like an environment in and of itself, right? So if a plant has a mycorrhizae interaction, could it suppress, inhibit, or prevent viroid infection? It could even facilitate viroid infection. Absolutely. In fact, there's a mycorrhizae-induced susceptibility, which is a phenomenon that has been documented where certain mycorrhizae will cause the plant host to become more susceptible to viruses, which is interesting. But of course, it's because resistance and tolerance and susceptibility, those are emergent properties. Those are results of genetic molecular interactions. So if those interactions change or are disrupted or are if they are facilitated, then those results change, right? That status changes based on those interactions at the molecular level. But at the same time, a microbiome can facilitate, the dynamic microbiome, the consortia of microbes can facilitate various interactions with these pathogens, right? So potentially they could, and I think that potentially with regards to the phytohormone production, a lot of microbial agents, both pathogens and mutualists, they do affect hormones and hormone production, because so much of that is tied into immune response and basic metabolism. So I could totally see how one or two or maybe even a complex of specific microbes might be able to facilitate certain defense compound production. But it's important to note that just because just because a collection of microbes may together for a particular host colonize, or rather they colonize the host, and they might be able to facilitate certain immune response functions or elucidate or elicit, not elucidate, if they're able to elicit the immune response or certain immune compounds or the production of certain, or the facilitation of certain signals that cause these terpene and other secondary metabolite production, that's great, but it doesn't necessarily guarantee that the plant's going to be resistant to a particular virus or bacteria or fungi or something like this. It's not a guarantee, but it could help slow down the pathogenesis or the virulence. Of course, the pathogens themselves also, and viroids in particular, they also will have their own interactions that will have to be contended, and it's potentially the case that you can still have a robust immune response, but it can still have sort of slits in the armor, so to speak, and that will cause a weakness that can be exploited, and that happens all the time. Can higher electrical conductivity or feed make plants more susceptible to viroids or viruses? I think I've seen some research that sort of shows that. It's kind of hard to answer that question because it's a very abstract effect that would be. Because, and I've also seen it characterized where people will say that bacterial pathogens can't exist between this pH or this EC, or fungi or viruses or that kind of a thing, but that's not always so true. I think that's kind of a little bit more complex than that. Certainly outside of the host, right? So inside, even outside the host, like the pH or the EC of the plant phloem, or the pH or the EC of the area outside of the rhizoplane is going to be very different, right? And yeah, so that could maybe have an effect as well. It's kind of hard to talk about it with too much detail, at least for myself. I think there was a paper about a virus viroid synergizing with botrytis or something. Maybe so. I do know that there are hypovirulence viruses that I've posted about personally with botrytis and other pathogens. So viruses can be beneficial for us and detrimental for even pathogenic fungi. There are several viruses, mycoviruses, that infect by botrytis and do not benefit it, and in fact cause it to be very ineffective at they get rid of the virulence factors, which is great. What do you think about John Kemp's plant health pyramid? I would like to see the sources for it. Personally, I haven't come across them, and I have looked for them, and I don't typically find them. I do think that the claims about plant health, kind of like what I just said, are a little bit too vague. Like, yes, it is definitely true that plant defense compounds obviously are defense compounds that have an effect on the pathogen or herbivorous insect that's feeding on the plant or something like this. But those organisms themselves have documented genes and enzymes that break down proteins and chemistries and things like this. Very, very commonly, a lot of herbivorous insects have adapted to them, either generalistically or in specialized groups of plants. An example that comes to my mind is how various native bee species, solitary bees, will they will go out of their way to collect these sort of like toxic chemicals and terpenes and esters and things, because they use them hygienically, and other Hymenoptera and other insects will do similar sorts of things too. They literally use these terpenes and things to clean themselves, or as a way to as like a pheromone even, or they're very important for sort of consuming them so that they get their own compounds for defense as well. Think of the monarch caterpillar, right? There's a lot of examples of organisms that will feed on a plant, and they will sequester the toxins inside them that are meant to hurt them, but they have no problem with it, and then they metamorphize into an adult, like a moth or a butterfly, and then they pollinate that same plant and benefit it with a pollination syndrome, or another plant for that matter. And yeah, so I don't disagree with the concept in general, but I do feel like saying that a plant can become so healthy that nothing will ever touch it is not supported by the ecological literature that I'm familiar with. Oh yeah, talking about the high Brix plants not being susceptible. Yeah, like a lot of insects do feed on the high sugar content in particular, or even the the nutrients, because what's nutritional for them, for the plants, is often also nutritional for the organism trying to feed on it. So that's it's not always the case, but a lot of times it is, and it definitely depends on the species, because some of them are just not adapted to certain plants, and for them, they're definitely going to have a hard time feeding on a non-host, so to speak. But if you're interested in more about that, I would recommend you check out my videos on YouTube about specifically about spider mites. They are really, really, really well adapted to their hosts, and different populations can become hyper specialized on different kinds of hosts. And I'm afraid this has probably already happened for cannabis, just as it has happened with other plants as well, when we've investigated their genomes and found that massive parts of their genetic code is for processing plant compounds. And they even have a few genes from bacteria, and I think even fungi, that allow them to break down plant defenses in a way that other organisms, other insects and herbivorous mites do not have access to, as a result of horizontal gene transfer. That those mobile genetic elements I was talking about earlier can have very, very massively effective disruptions to those ecological interactions. So just an interesting thing to consider, and is one of the reasons why I find that making really simple, broad statements about ecological interactions tend to sometimes mislead us into thinking that we are more defended than we actually are. And yeah, so I think that'll be it. I really appreciate the conversation with everyone. I'm glad that I had a lot of people asking questions about things like exosomes and the hormone pathways and sort of other esoterica related to plant health and plant physiology. So that's something I'm very interested in, obviously. And if you want to see this video again, you can check out it on the IGTV. You can also find it on my YouTube channel, Zenthanol, where I will probably also post the research documents in the video itself with a little bit of editing, or at the very least in the descriptions for further reading. And if you have any additional comments or questions, please leave them in the comments below and share this video with people who you think will benefit from the information about viroids. Hopefully it was illuminating to everyone here. And I was happy to see the support of so many people who are interested in the research and empirical based literature about viruses and viroids in plants. And I'll probably make a lot more videos about these in the future. Hopefully on YouTube, but definitely on here too. Have a good one.